this it? Awesome. There we go. Okay, we're going to talk about basics of DNA structure. Put together a little model with tubing, tape, and pens. So you can hold it up. See if I can use it to illustrate what I want to illustrate today. So those of you watching on the camera on YouTube, if you could give me some feedback as to whether you're able to see the screen and whether you're able to see my writing, I may need to come up with some other solution to this. For the students who are here in class, just to remind you there are a couple of students that have a conflict, and I've been a nice person and just signed an override form. I'm doing an experiment. We're filming the class this semester. They're responsible for posting that on YouTube. They are going to send me the account with the link, and I will send that out to all the students in this class so that you'll be able to go and look on YouTube and view the video of the classes. So if you're out for, you have running or track or cross country or something like that. I know the students all have a lot of activities. Some of you are seniors and you'll be doing interviewing. Yeah, interviews are important, it's okay. I understand you miss classes, but this is your opportunity to catch up on it. So, starting off with nucleic acid structure. Probably a lot of this is a review. Pardon me. From General Biology 1, I want to define the terms as I will be using them through the course of the semester so that we have the same starting point. Sometimes I use words a little differently and I describe how molecules are used a little differently. I just want you to start to see how I frame things, put it together and package it in my head to understand. Hopefully you guys will be able to also. So in general, there are two types of nucleic acids. As far as life on Earth, there's the RNA world and there's the DNA world. And you should know DNA is composed of 4 nucleotides, A, T, G, C. And RNA is also composed of 4 nucleotides, A, U, G, C. And those are the parents, A and U. There's a paper that was recently published in which they suggest the original life on Earth, whether it was a pre-cell or an actual cell, was likely an RNA genome based, and the nucleotides that were used were not these same nucleotides. These nucleotides came into use after this pre-life or early life. They've been around for a while and just adopted using these chemicals because it's more stable. Pretty interesting stuff. We're still learning a lot. Obviously that's all hypothetical. So, if I draw this out, and I'll draw it out in a real generic structure, and this is what I'm looking for from you. I'm not looking for you to draw out every single atom in the proper conformation, right? So basically what we start with is we have a ribose sugar, which is a 5-carbon sugar, and there are OH in groups. The simple definition of a sugar is a string of carbons on which there's an OH poking off of every carbon. Okay? So this carbon here, this is the OH group for that. I think. No, there must be an OH here. The OH, this carbon has this as an OH. There's an OH here, there's an OH here, there's an OH up here. The OH for this guy is over here. 5 carbons. If you remember your organic chemistry, if you've taken organic chemistry, there is a way of numbering the carbons. So in a molecule with 5 carbons like this, there is a precise, the chemists have come up with a precise definition as to how you determine which carbon is going to be called carbon number 1. And I think that definition is loosely defined as the carbon with the most reactive or most important group hanging off of it is carbon number 1. Any chemists here who would agree or disagree with me on that?
Sounds right.
Sounds right. Loose. Yeah, that's for the purposes of my biology class, that will do.
That works.
So this is called carbon number 1, this guy right here. And the reason is we connect onto this. This is what we connect the base onto. And the nitrogenous base is going to be an organic molecule with a large amount of nitrogen in it. It can have a single ring or it can have 2 rings. So it can be one ring or two rings is the way I draw it. Adenine and guanine have two rings. Thymine and cytosine have a single ring. Right? And it winds up that these guys do have two hydrogen bonds and these do three. We'll talk about that later. So if that's carbon 1, then carbon number 2 is located here. Carbon number 3 is located here. Carbon number 4 is located here, and this is carbon number 5. So ribose with a nitrogenous base. I think this is called a nucleoside. I don't have it on here, it's on the next screen, I think. If you add phosphate groups and you basically charge some energy onto this molecule, make it a higher energy molecule, you put phosphate groups on here. So if we were to make this nitrogenous base, let's say we're going to make that adenine, and I have a single phosphate here, what I have is adenine monophosphate. If I add another phosphate group covalently connected to this one, I'd have adenine diphosphate. Is this an important molecule in the cell? Yeah, AMP is important. ADP is more important. If I add a third phosphate on here, obviously we get what I refer to as the money of the cell, the currency of the cell. That's the energy that gets things done, right? You think, oh, you just burn some ATP and then you make such and such happen. You learn all that, right? The reason this is such a great energy molecule is each one of these phosphate groups is negatively charged. This negative charge does not want to be next to these guys. And so when it's very eager to leave, and there's a lot of energy in its release, it's a very high energy bond to hold it in place. It's a great energy molecule. It's not very stable. So this is ATP, adenosine triphosphate. This is a nucleotide. Had anyone prior to today, had anyone ever heard the word nucleoside? A couple of you, probably in biochemistry course. Yeah. If you add a phosphate on, now it's called a nucleotide, right? So what we have here is actually a nucleotide that's used in the RNA world. What would be the change if I were to convert this into a nucleotide that's going to be used in the DNA world? You know, I said A is used in DNA and A is used in RNA, but there actually is a very subtle difference. What's that difference? The deoxy.
You have the oxygen on number 3, right?
Nope. Close. Right. Number 2. So this is an adenine that would be used to build RNA if this, instead of being a hydroxyl, that's an OH here, especially for those of you on the camera, there's an OH. If I were to convert that hydroxyl, into just a hydrogen, I have taken an oxygen off, and now we call this deoxy ATP. And we note that with a small d in front. Deoxy ATP. I guess hypothetically that could be used as an energy molecule in the cell. I don't know, probably someone has tested that out, and I don't know if that actually happens. When we talk about ATP in the cell, we're talking about the RNA form of ATP. That's the energy molecule. And in general, you don't want deoxynucleotides around because they can be used for DNA replication. You want DNA replication to be performed only at specific times. We'll talk about that in cancer biology portion, of course. Okay? So this is about as specific as I'm asking you to be able to draw these things. You can just put like a circle or a double circle up here, or NT or NB for nitrogenous base, whatever you want to do. Okay. So we've defined the components of a nucleotide. We've defined the difference between deoxy and normal ribose nucleotides. So, is this deoxy or is this deoxyribose or is this normal ribose? Is this DNA or RNA? RNA. This is RNA. Because on the 2 carbon, I have an OH. Okay. So on the exam, if that were question number 1, you'll be able to get that one right for me? Good. What happened there? Okay. So, we can actually covalently connect these guys together. And that happens if I have another nucleotide coming in. I'm not going to draw all the OHs on it. We've got some functional group. We'll go nitrogenous base off here. We'll put 3 phosphates moving off here. When I draw a P with a circle around it, that means one of 2 things. It either means Paul Og, that's how I draw my initials, or it means phosphate group, PO4 with a minus charge. I think it's standard across biology and even chemistry that this means phosphate group. Okay, so that's how I draw it. What I can do is I can actually react this phosphate. I think it does a nucleophilic attack on this bond, such that a bond is formed, that the bond that used to connect between these 2 switches and connects over here. It breaks this bond and it breaks this bond, and we release a phosphate group connected to a phosphate group. Does anyone know what that's called? What? No, what's this thing called? What's that chemical called? Does anyone know? Pyrophosphate, yeah, awesome. A little bit of trivia. It won't be on the exam. You look good in front of you. You look good on the camera. Maybe at the end of the class, we'll shine it over on you, okay? Okay, so what happens is we've released, that's a tremendous amount of energy that gets released, and I've covalently connected this to this. Am I doing this right?
It attacks number 3.
Oh, yeah. Totally wrong. Totally wrong. It's this bond. Yikes. We can edit that out. It's the magic of television. Sorry, there's this probably does a nucleophilic attack on those bonds. But this bond happens. Thank you for that. A lot of people were looking at me like I was a crazy man. And you were right. A student told me that I have a caveman beard. To which I said, me Og. That's funny. You're clever biology professor. Okay, that's right. So the beauty of the situation is we've got another OH here that can be available. If I brought another nucleotide in, I could perform that same reaction. Right? So we can continually add on to this number 3 carbon, because this is a new number 3 carbon here. Number 3 carbon, number 3 carbon. We can add another nucleotide onto that 3. Okay. To the best of my knowledge, there is no way of adding a nucleotide onto the 5' end, onto this side over here, with one bizarre sort of exception, which you couldn't say is really adding onto the end. We'll talk about it later in class. So this is all illustrated here. Here, yeah, I've correctly illustrated. This pyrophosphate, oh, here's, did you read it off there?
There you go.
Oh, great. So the pyrophosphate is released and that covalent bond is now made up here. Okay, great. So here we actually have reacted this nucleotide on and then this nucleotide on, this nucleotide. So we released, what, 2, 4, 6 phosphate groups, or 3 pyrophosphates process. And again, we still have a 3' hydroxyl down here that we can add on another nucleotide. So if we now look at this bigger structure, this creates, and if you haven't heard it by now, you have to learn it. I'm going to start using this as just your general term. I'm just going to throw it all around class. We're going to rely on this as a fundamental concept. There is a sugar phosphate backbone. I have a sugar, I have my deoxy ribose sugar, so this must be DNA, connected to a phosphate group, connected to a sugar, connected to a phosphate, to a sugar, to a phosphate, to a sugar, to a phosphate. This is a long chain. The largest human chromosome is 200 million base pairs, which means this sugar phosphate unit repeats 200 million times in a row. That is one huge organic compound, right? Poking off of that sugar phosphate backbone every space periodically is a nitrogenous base. Every ribose, unless the DNA is damaged, every ribose has a base. In your body, probably every cell has got a mistake in it where this thing is like accidentally kind of floated away. And now your body will need to come in and repair that, hopefully correctly, sometimes not. And so you're continually moving towards, unfortunately, towards a state of cancer. We'll talk all about this as the final section of the course. So, sugar phosphate backbone, you have to understand that sugar phosphate backbone concept. With these guys just poking it off. They're along for the ride, right? Any questions so far? Okay. Last point on here. Each one of these phosphate groups is negatively charged. So this negative charge and this negative charge are the best friends, right? No. They want to be the hell away from each other. So what we've got, if I had a single strand of DNA with all my bases poking up like this, the negative charges on each one of these want to be as far away from each other as possible. I want you to imagine, let's say I could hypothetically, I could take something that's got a really strong positive charge, and I were to put that thing right here. Well, we're not going to put just a few of them in here. How is if I were to force this to be in close proximity to the sugar phosphate backbone of this DNA molecule, what will be the effect on the DNA molecule? I've neutralized the charges. What happens here? I have some object. It'll twist, it'll wrap around. So if I had it in its normal state, with all these negative charges on it, my ability to bend this thing back and forth is very limited. If I bend it this way, I move these guys close to each other and they don't want to be there. But if I neutralize those negative charges, now this DNA becomes flexible there. I can bend it all around. In fact, if I put 4 positive charges there, it means I have a partial positive charge left over. And I neutralize 3, so this negative charge will actually distort when I move and hang out close to that. I can take something which is very rigid, just by neutralizing the charges in the DNA phosphate backbone, I can get that thing to bend around a charged object. Like what? Histone. And histone is a protein that has a lot of positive charges. Transcription factors. Okay. This is in red. Why? It's important. I almost never put text in red. So, the same way that chemists said, hey, we're all going to sit down and figure out how to name, why do we decide this is carbon number 1? And they all said, well, we're going to come up with a rule, and any chemistry student is required to learn this rule, so everybody will follow the rule, and we all know things will be numbered properly. Because you guys have all learned it, and you'll never forget it, right? A bunch of biologists got together and said, well, if I have this sequence here, I could write it TACG, and the guy in the lab next to me could write the sequences GCAT. We want everybody to write it in the same order, the same way. So we're going to say, if you write a nucleic acid sequence, a DNA sequence, or an RNA sequence, by convention, by definition, you will always write it from the 5' to the 3'. You write the sequence from the 5, this is the 5 carbon right here. You start there and you say, what is the nucleotide attached to this? What is the nitrogenous base? Sorry. That is the first one I write. And then I go down to the 3 and I come to another 5 carbon. What is the nitrogenous base attached to that? So that here I have my phosphate group, and here I have my 3' hydroxyl down here. By convention, this sequence must be written this order. What is the correct sequence of this? TACG. TACG. So, pardon me. If you see this sequence in your brain, I want you in fact, if you see this sequence written just like that on a piece of paper for the purposes of this class, while you're in this class, I want you to write a 5 over here, and I want you to write a 3 over here. If there is a sequence which is unlabeled, you are to assume that this is the first nucleotide moving from 5' to 3'. Okay? Questions on that? So this is Linus Pauling, and he worked with proteins. And he proposed that proteins were these flexible, basically these flexible little cords of amino acids, and that these cords would roll up and form these secondary, tertiary, quaternary structures. They have a 3-dimensional shape to them, the way these threads would fold up. And it turns out, people who were studying DNA said, DNA must do the same thing. DNA must fill some 3-dimensional space, and the way we think of how that fills space is going to follow the model that was defined by this fellow. And so here is actually, this is an amino acid sequence which bends all around, and there's some hydrogen bonds that form that hold this into a particular shape. You probably know this by now as alpha helix. You should have learned that in Bio 1. We can come up with, so the amino acid sequence as you would write it out on a piece of paper is what we call the primary sequence. How those things interact with each other when they're the amino acids right next to each other, how they interact with each other is the secondary structure. This happens to be an alpha helix. How an alpha helix and an alpha helix find each other and interact with each other is a major fold of the protein. It's tertiary, and quaternary is if you have 2 different proteins come together and interact. And we can follow that exact same pattern with DNA. We'll see that at the end of today. So trying to determine the 3-dimensional shape of DNA, you're probably familiar with Watson and Crick. They earned the Nobel Prize for elucidating the 3-dimensional shape of DNA. They based their model off something that already existed. It was not really original thought on their part. The data that they obtained that allowed them to figure out the structure of DNA was actually conducted in a different laboratory, and they had about a 5-second view of it. The woman pulled out her little data, showed it to them. From the data, they went, oh, that's another piece that I can use to fit it all together, and they figured it out. That person is Rosalind Franklin. And there's some controversy behind, oh, did they like steal the data and run off and like, screw you, we're going to publish this. And, I wasn't around, I don't know the details on it. In general, researchers are a little protective of data they haven't published yet. She hadn't yet published this. They saw it publicly. They acknowledged the importance of her data, but she wasn't an author on their paper. And if you're going to win the Nobel Prize, you need to be alive. It's not awarded to dead people. And I think she was not around when the Nobel Prize was awarded. My guess is if she was around, she might have been awarded. It was a different time in the role of male and female in this country, in general in society, so I don't know if that would have impacted this or not. Anyway, so she had a piece of data, and based on what they saw, they realized the structure of DNA needs to be some kind of a helix. It needs to basically be something that spins around on itself like this in a spiral. And data based on that another researcher had put forward is, if you have, say you have a bacteria, and you break its DNA apart to see what % of the DNA goes to A, what % percent is composed of T and what percent is composed of G and what percent is composed of C. If 10% of the genome is A, always 10% of the genome would be T, which leaves 40% for C and 40% for G. If you had a different organism where if 30% of the genome is A, always 30% of the genome would be T. Anytime you had a percent that was A, the percent that was T matched almost perfectly. And it didn't matter if it was a virus, or if it was bacteria, or if it was a mouse. That relationship of A and T always having the same amount, and G and C always having the same amount was a clue that these guys interact in some way. They're paired up somehow. So the first model that they came up with was to basically have a charged molecule that could neutralize the negatives of the phosphates. And the phosphates from the sugar phosphate backbone would be attracted basically to a string of, in this case, magnesiums, and would hold the strands together, and then the nitrogenous bases are poking out and they can just easily be read, and you need to read your code in order to express things out of the genes. So that was really sensible. And they tried, and the width of this was too wide based on Rosalind Franklin's data. I think it was Rosalind Franklin's data that said the DNA molecules are this wide, it's only this wide. If they took just a single strand was only this wide, so it had to be two strands. But how they came together, they didn't know. So, Watson took some scissors, an old cardboard box, and he drew on them to scale, the size of the atomic bonds that form all of the covalent bonds in A and T and G and C. And he had the hydrogens and the oxygens poking off, and he just sat there with the shape that he'd cut out that was A, trying to figure out how it would interact with T, and he found a spot where they would line up and form a really beautiful hydrogen bond interaction. And then he took a cutout of C and a cutout of G, and he lined those guys up and found another beautiful hydrogen bond interaction. And the incredible thing is, when he had A and T lined up, he could take G and C, and he could take these nucleotides, he could set those cardboard cutouts on top of these, and they filled almost exactly the same width, exactly the same space in three-dimensional space. So he figured, all right, this is how it is. They match up this way. And I must have one strand goes this way with nucleotides pointing in this way. One strand goes this way with nucleotides pointing in this way. And the nucleotides interact and hold themselves together. And the width of that almost perfectly matches the width from the X-ray diffraction data. The problem is, if you take a bunch of these, I'm going to draw them like this. So we're looking sideways on the cardboard cutouts. To get them to line up, the sugar phosphate backbone had to do some crazy stuff like weird angles, like impossible angles, to get these things to line up in a nice row like this. And this is where Watson just couldn't move forward, couldn't figure it out. Well, this is my understanding. My understanding is that Crick came along and said, boy, that looks really interesting. Why don't you take this strand and turn it around like that and see if that works? And so that's what Watson did. And the sugar phosphate backbone was a nice clean line in both cases. No stress on the bonds. There's very little stress on the bonds. Questions on this? Kind of cool, right? So this is a very famous image. It's probably in most probably all introductory biology textbooks, every genetics textbook. This is a picture of Watson and Crick. So they're looking at this ball and stick model. Most of that ball and stick model still exists. Some people have actually, before it became like some real sacred things, people nabbed off bits and pieces of it and would occasionally call and say, hey, would you sign my nucleotide? And apparently, towards the end of the career, we're not too happy about it. But anyway. So, here are the key features of DNA. This is an important slide to keep in mind as you're preparing for exam 1. These are the characteristics of DNA. The sequence of one strand is complementary to the sequence of the other. If I gave you the sequence of one strand, you could automatically tell me what the opposite, or the compatible, the complementary nucleotide is over here. You learned that in Bio 1, I hope. The bases form hydrogen bonds. A and T form 2 hydrogen bonds. G and C form 3 hydrogen bonds. Practical relevance of this is, if I have a piece of DNA, and there's a long stretch of A's and T's, and I have a different piece of DNA where there's a long stretch of G's and C's, my ability to pull those apart is different. Because the A's and T's have fewer actual hydrogen bonds holding them together. The G's and C's have 1/3 more, 50% more hydrogen bonds holding it together. A and T separates more easily than G and C. Mammals, if you look at genome, the amount of G and C in the genome of an organism. Frogs and reptiles have, I'm going to make a number up here, say 35% GC. Their genomes are mostly AT. In mammals, we have about 50% GC or more. And some would say that we have warmer bodies, and to keep our DNA together, the DNA had to evolve to have more GC. Sorry, I should go anti-parallel. Does that make sense? Which is kind of cool when you do these genetic analyses. The two strands are parallel to each other, but they're anti-parallel. What does the anti-parallel mean? And it comes back to how we write this out. If I have one strand here, and I write this out by definition, you should already be thinking to yourself, I'm supposed to write a 5 here, and 3 here. This is a 3' hydroxyl, and this is a phosphate group hanging on here. On the end, that's the end of the molecule. The other one is parallel. Well, we know it's complementary. But it's parallel and complementary, but it's anti-parallel. The 5' end of this guy is here, and the 3' end of this guy is here. So, wow, what a challenge. What if I write a sequence out, and the important sequence, let's say this isn't the strand that's used. Let's say this is the strand that's used. And the important sequence to say is GGCG, this part right here. How am I going to illustrate that? And the answer is, does someone want to give the answer? Do you know? It's simple.
What are you asking?
So let's say the DNA, this sequence of DNA has some important function. And it's these nucleotides that are important. It doesn't matter what these sequences are up here. So this is the important strand. If I want to talk about this sequence here, I wouldn't write it this way. How would I write it? Would I write it like this? Would I write it that way? No. What I would do is I would rotate it this way. I hate doing this. I have done this thousands of times in my life. I've hated it every time. Where the hell am I? Did I do that right? Yeah. And then I'd write this one underneath it. And then I would note this somehow as the important stuff. sense to you guys? Okay, I need to hurry up here. Okay. The backbone is on the outside. Let me get into my little model. We'll see if I can make this work. Oh man, it's so folded up now. I'm not sure I'm going to do this mess. There we go. Scotch tape and pens. Oh, it's breaking already. I should have brought my tape. These represent the pens represent the nucleotides. This is the sugar phosphate backbone. Okay. So the DNA, in order for these bonds to work, the DNA actually needs to twist around a little bit. So what I want you to see is, I want you to follow this space in here, and I want you to follow this space out here. As I spin this around, the space out here becomes wrapped into this thing here. Does that make sense? I capture that. And you can see I have a nice like spiral staircase going in here. So, this nucleotide here is hydrogen bonded to this nucleotide here. There are two nucleotides, two nitrogenous bases interacting here, right? There is absolutely, on Earth's pressures, there is no way to push this closer to this. This cannot be pushed closer to this because these chemicals are physically in the way, right? This is called the minor groove, this distance here. Now, in addition, there is another groove. If this is the minor groove, this space here is the major groove. I'm going to show that to you up here. This is the minor groove, and that's the major groove. See how this is larger than this? This is smaller than this thing here. You guys understand that? The minor groove cannot change in its size. But as I curl this around and this space gets wrapped in here, if I wrap this more tightly and put more spins on it, the major groove can get crunched and shrink smaller and smaller. I don't need you to spatially understand all that. I need you to conceptually understand. The minor groove is smaller, and almost always smaller than the major groove. Okay? Some people incorrectly, and I've seen this in textbooks, some people incorrectly say that the minor groove is this space and the major groove is this space. And the reason it's incorrect is then that the major groove would be the same size as the minor groove. Right? Because you're just saying two opposite faces of the same distance. So the major groove originates from space outside as this gets spun around. Okay, that's what I want to get across conceptually. This is really, really important to me. One of the reasons I put that Linus Pauling picture up there of the alpha helix of a protein, proteins will have amino acids fold into an alpha helix. It's a very common three-dimensional shape of many proteins. The shape of an alpha helix is too big. It physically cannot get into the minor groove. I need to work out. An alpha helix is too large to click into that space. But an alpha helix is the perfect size to click into this space here. So that the ionic interactions or the hydrogen bond interactions that this thing could potentially make with the DNA here could make a very specific interaction between one protein and DNA, and all the other proteins in the cell could not make that interaction. I can control whether that protein can interact with the DNA or not by controlling how compacted or how twisted the DNA is. If I loosen up the DNA, I can open this up, and maybe some other new protein can get in there that couldn't before. And the cell can control whether that second protein interacts or not by then twisting the helix more tightly and squeezing this space. How cool is this? So the major groove and the minor groove, this is a structure that we call, the way this is laid out, we almost always think of DNA as being B form of DNA. And that is, if DNA were relaxed and able to take on its natural shape that the covalent bonds were directed to do, it would have a twist of 13 bases per twist, or 10 bases per twist. I can't remember what the exact number is. 8? 10? 10 bases in one full spin of the helix, yeah, something like that. So that's your typical B DNA. If we twist it more tightly, we call that A DNA. We'll talk about how DNA can get more twisted more tightly. It takes a force. You have to apply a force. Because the covalent bonds of the DNA don't want to be in that conformation. And to distort the shape of those bonds, you have to apply a force and keep that force applied. We'll talk about how that can happen later in the semester. Well, we'll talk about numerous points. Hopefully, if you're paying attention, you'll pick up on it and go, oh, okay, that's how it happens. And when I finally get to the big aha moment, you've already been there, but you do that already. You could actually take DNA, instead of spinning it this way, you could spin it the other way. You could untwist it so much that you twist it backwards the wrong way. Boy, that would take a tremendous amount of force to keep it in that conformation. We call that Z DNA. I don't have time to talk about how, but researchers have demonstrated that Z DNA exists in your cells. Your cells intentionally wind the DNA backwards, apply a force and maintain it in a distorted shape for a very good reason. This is going to blow your mind. DNA is a two-stranded helix where the strands run antiparallel. There exist organisms that have three-stranded DNA. It's not the entire genome, but they have portions of their genome that are double-stranded, and a third strand of DNA can go in and interact. It sits into the major groove and wraps around, and it's not just like randomly goes, hey, I'm a third strand of DNA, I think I like to bind here. Let's make the nucleotide sequence, the bases, the order of the bases in this third strand determine whether it can interact with functional groups in the major groove. And so it's sequence specific. If we come back to Earth in 1 billion years, we may see that Earth has been taken over by organisms that have three-stranded genomes. And the entirety of their genome is three-stranded DNA. It may be the next huge evolutionary step. I'm not sure if human DNA does this intentionally, but there are organisms that do intentionally do this. But the value of this is, let's say I get infected with HIV, human immunodeficiency virus. That virus can actually break my genome apart, insert itself, and then seal itself back up, and is now a part of my genome. And I could be clear of the virus and have no sign of the virus, and then 20 years from now, the virus cuts itself out of my genome and then starts an infection all over again. I could potentially prevent that from happening if I were to take a pill, say, and that pill delivered a specific sequence of DNA. That DNA would penetrate into my cells, and it would interact and insert itself into the genome of the HIV virus in my genome and prevent the virus from ever coming out. There are ways we can target and shut down or activate parts of our genome using this third DNA strand model. And this is very real research out there. People are when people make patents on this stuff, there's going to be billions of dollars to be made. Okay, I've talked about Linus Pauling and the protein structures. So the three forms of DNA. The primary structure of DNA is the sequences you would write it out on a piece of paper. The secondary structure of DNA is the double helix. The tertiary structure is how that DNA folds upon itself. Pardon me, and the quaternary structure is different DNAs interacting with each other, which can sometimes happen. So very sensible to what we learned about protein structure in general biowork. RNA, also, usually in our cells, RNA is single-stranded. But that single-strand RNA can fold up on itself, and you can actually see this great example. There will be sequences in an RNA, even if you just randomly came up with a sequence, where the sequence can actually fold back on itself and hybridize with itself and satisfy the hydrogen bond with the base pairing. So here, these sequences match, and then they don't match, but these sequences match. So this is called a stem loop. So this is secondary structure. If I were to draw those possible interactions out on a piece of paper, this is secondary structure. Tertiary structure is how these stem loops all fold onto each other to make a three-dimensional shape like this. And this should be familiar in appearance to you. This is the transfer RNA. And this stem loop is the anticodon that reads the messenger RNA and knows which amino acid should be brought in. The amino acid is connected to the 3' end up here. So if this goes into the ribosome and this matches with the codon that's there in the ribosome, it will sit down and transfer the amino acid onto the chromatopeptide chain. We learned about translation in more detail in this section of the course. Okay. So we're done with class. I want to take, well, we're finishing in 2 seconds. I want to take 15, 30 seconds to talk about my exams. Some of you have had my classes and you know how straightforward my exams are. There's no tricks. Some of you have not had an exam with me before. I want to explain to you how I write my exams. I will take this slide and I write every exam new and fresh each year. I will take this slide, since this is lecture number 1. These are the key points. If you can do these, if you do every one of these from every slide, every key point slide, you should get 100% on my exam. When I sit down to write the exam, I will look at this key point and I will try to write a question. So, I guarantee you that question 1 on exam 1 will test this concept. I guarantee you that question 2 on exam 1 will test this concept. Question 3 on exam 1 will test this concept. I am not one of these professors who says, if it's in the book, if I talked about it, if I alluded to it, if it's common sense, it's on the exam. I don't do that. This is the exam. This is it. The questions assume you know these words. If you don't know these words, you're not going to be able to understand the questions that I ask you. You're on your own on the exam. People who have taken my class before, is that an accurate description?
Yeah.
I don't think you need to buy the book. I make this after I write my PowerPoint file. If it's not in the PowerPoint file, it's not on here. Okay? Great. Have a nice weekend, everyone.